Okay, these are the notes from today. This is the first part of Chapter 3 in biology, and it's just basic information leading into the organelles and into the different types of cells. So again, we all know that our body are multicellular organisms that were made up of more than one cell. And cells in multicellular organisms are differentiated. Or they have different types of cells, which all have different functions. Again, think of our own body. We've got skin cells, we've got blood cells, we have bone cells, muscle cells, all kinds of things. And each one has its own specific function, although they all have basically the same parts. Again, we're going to call this specialized or differentiated. Okay, how we study cells is one of three ways. Generally in class, we'll use what are called compound light microscopes. Compound meaning that it has more than one magnifying lens. In this case, they have a lens in the eyepiece and also in the objectives down here. And the nice thing about the compound light microscope is that we can use them to examine living cells. One of the disadvantages of them is that light must pass through the specimen, which means that the specimen, or what we're looking at, has to be really, really, really thin in order for the light, which is down here on the bottom, to penetrate so we can see what's on the inside. Another type of microscope, and we'll play with these a little bit, it's called the dissecting microscope. These have lower magnification, and they provide a three-dimensional view, which means the light is coming from up here instead of coming from the bottom, as we saw with the compound light microscope, which means we can look at things that are thicker, and also, like a fly or a piece of a leaf or something like that. So it gives you some more dimension, but not as, again, it's not going to be as detailed as what we see with the compound light. The third type of microscopy is the electron microscope, and you can tell just from the picture that this is going to be a very expensive machine. You can see all the computers it's linked to, but the really cool pictures in biology books come from electron microscopes. Now, like the name implies, it uses a beam of electrons in order to create the image. Also happens in a vacuum. Problem with this is whatever you're going to put in there is going to be dead, because the electrons in the vacuum is going to kill the specimen. So you found something that was new and exciting, like a single-celled organism, and you wanted to try to identify it and find information about it. You would not want to use an electron microscope because you just killed your very unique specimen. Or if you was looking at a cancer cell, wanting again to determine type of cancer, you would want to not use an electron microscope necessarily unless you had a bunch of cells to work with. So a little bit of history of microscopy. All of this started in about 1665 by a guy by the name of Robert Hooke. And he used his microscope to view larger single-celled organisms such as protists, algae cells. He looked at sperm cells, and he looked at cells from cork, which is just a type of tree. And he coined the word cells because, again, we take cells for granted. We've just grown up with it, but somebody had to decide to call them cells. And he coined this because the cork cells reminded him of the cells or the small rooms in a monastery. Again, think back to this time. Monasteries, nunneries were very, very popular. A lot of people lived in them. And again, the rooms are very much like a prison cell. Again, that cell word coming up. This is a slide that you've got to know. Cell theory is made up of three basic components. First of all living organisms are composed of cells, are made of cells. Again, we kind of take that for granted, but that wasn't always known. Part 2, cells are the basic units of structure and function of living things. Seems an awful lot like Part 1, but we're kind of splitting hairs here and kind of looking at things a little differently. The third one is that new cells come from existing cells. Again, this is something we kind of take for granted, but at one point, people didn't understand where cells came from, and they thought they just appeared magically or spontaneously. And at this point, we know that that is not true. We have two basic types of cells, and again, hopefully from your drawings, you realize that. The first is the prokaryotic, or the bacterial cell. These are very small. They're 0.1 to 5 micrometers, and a micrometer is like it's 10 to the negative 6th of a meter, or 10 to the negative 3, or 1,000th of a millimeter. So again, we're talking something that's really, really small. And they're small enough that they can use diffusion to move materials in and out. The most common type of cell found on Earth is a prokaryotic cell. In fact, our body has more prokaryotic cells inside of it than we have cells in our body. These are going to be simple, they're unicellular organisms, and they have four basic components. And again, this is something you really need to know. We have the plasma or the cellular membrane, we have the cytoplasm, we have the genetic material, and we have ribosomes. The second type of cell is your eukaryotic cell. And these may be unicellular or multicellular organisms. They curve either way. The size ranges from 10 to 100 micrometers, so again, significantly larger than the prokaryotic. But that cell can't get too large because if it gets too large, we can't get materials in and out of the cell efficiently. And again, as we know from our drawings, eukaryotic cells have differentiated components that recall organelles. So the mitochondria, the Golgi apparatus, the ribosomes, all of those would be considered differentiated components. And again, they all have their own special function. So how did eukaryotic cells come about? These guys came in after the prokaryotic cells. The prokaryotes were first. And the theory is endosymbiosis. Now, endosymbiosis is where two organisms or two things benefit each other enough that they work together. And what happened is individual cells that lived in similar environments eventually merged into one cell because maybe one was making one type of energy and maybe one was providing some protection. And they decided that it was better to live together than to live apart. And there's lots of supporting evidence that to confirm this. One is that chloroplast, the mitochondria, and the prokaryotic cells are similar sizes. They also all have a double outer membrane, which you do not see in your other organelles. And they also have chloroplasts and mitochondria that contain their own DNA. This is where the maternal DNA comes from when we're talking about genetics. This is the end of this first section. These are some things that were different and new that maybe you hadn't seen before when you did the cell structure. And it starts with junctions between the cells. We think of cells being lined up as like bricks, but it's not necessarily that way. In plants, they have what's called plasmodesmata. So plants, plasmodesmata, are channels between the cells. So again, the cells are able to communicate. Animals have three types of junctions. They have one that's called tight junctions, which there's no movement of material between cells. And this is really important, like with the stomach and with the intestines, where we really don't want stuff leaking out of the stomach or the intestines into the rest of our body. We also have what's called desmosomes, and these are where the cells are tethered, kind of like a boat on a dock. You want to tie the boat up so it doesn't drift away, but you want it to have some looseness so in case a wave comes through that the boat's not torn up. Desmosomes kind of work that way. They kind of tether cells together. Good example of this is your skin cells. If our skin cells were tight junctions, if we pushed on our skin would shatter. And obviously we don't want that. So we want some connection, but not something that's super tight. The third type of animal junction is called gap junctions. And this is channels between the cells, kind of like what you see with the plasmodesmata, and it allows for information to be conducted between the cells quickly. And a classic example of this is our heart, because our heart, all the cells have to pump at the same time. They don't, the fluids get backed up and then we run into major problems. So again, gap junctions allow for quick communication. And the last slide from today was the extracellular matrix found in animal cells. Again, extra means outside as opposed to intra, which is found inside. And again, so material found on the outside of the cells. And it contains glycoproteins, protein collagen, and fluids. Again, think of like your knee. You've got fluids in there. It's not part of the cell. It's not part of the tissue, the muscle tissue and stuff, but it does serve a function. And again, this extracellular matrix provides support, protection, and allows for communication.